As we move here towards finishing out everything, I'd like to invite whoever is going to be presenting from each one of the breakout sessions to go ahead and come on up here. And yeah, I'll just, if you'd like to go ahead and start, Jordan. Sure. We've got cardio up first.
Efficiency is important, right? So I want to thank everybody for the opportunity to come here today as part of my role at the FDA in the Division of Cardiology and Nephrology. I was not aware that this existed until a few weeks ago. The DARS folks reached out to me looking for somebody with clinical experience. Just so happened that I had a lot of relevant academic experience in tissue engineering in the cardiac space. That is very nice to fall back on as we had this discussion. So we had a lively conversation about how best to design a context of use statement. We really identified a lot of areas of unmet need within the cardiovascular space to help guide that discussion. There's robust information in the cardiac conduction space about using microphysiologic models to model risk. And we heard that from Ksenia Blanova today, especially using it to do QT, QTC, QRS, less known as kind of atrial effects, PR prolongation, AV block, just general chronotropy. Can we predict heart rate effects? Inotropy, can we look at both positive and negative inotropy of drug candidates with CIVMs? And then, the one I brought to bear today was really cardiotoxicity and understanding whether these microphysiologic models can be done on a cellular cytotoxic or direct effect on the contractile machinery. And so questions of interest that are subcategories to this are, of course, special populations, which are of interest to the FDA in diversifying our clinical trials and understanding about specific effects in these special populations. Combination therapies, especially within the oncology space, where known agents that have cardiotoxic risk are being added to new therapies with unknown if they'll have additive or synergistic or even improve the outcomes there. Acute and chronic adverse effects, modeling both are important. The chronic ones may be more challenging, but we heard today that there is possibility. And then being able to turn reversibility, looking on and off rate kinetics. Complex cell-cell interactions, of course, we want to recapitulate the disease or pathophysiology or biology of interest, and so having at the right cells to your in vitro model is important. And then, of course, for what we're really doing here is relevance to preclinical to clinical translation or de-risking a drug once it's in early phase clinical trials. What we also wanted to iterate is that models to actually do that or to begin to think about this exist, so we can actually start to get some movement forward in this area. We took the time because cardiovascular, the vascular piece of this is often commonly added to CV for cardiovascular disease. And so we talked about, can we model blood pressure with CIVMs? Can we look at the components of the physiological equations for the heart, cardiac output, systemic vascular resistance that lead to the eventual blood pressure phenotype that you see with a cuff? And so putting vascular components, cardiac components, and not to be forgotten, Elisa, if she's still here, the kidney components as well. And then, of course, vasculitis, drug effects on the vasculature, intravascular hemorrhage or clotting, which is very much dependent on the substrate of the vascular system, the endothelium as well as smooth muscles. And then we heard a specific use case about coronary arterial hypertrophy seen in preclinical animal modeling. And again, we have the same kind of questions of interest, especially subpopulations and combinational therapy. We chose to focus on the cardiac space, and really that's because there are not well-developed or characterized CIVMs yet to address some of these blood pressure questions, and so that's somewhere that we can think about for a future session possibly. Okay, so let's get to our context of use and thinking about unmet clinical need for a CIVM in the cardiac space. And so we really thought about it in the prospective toxicological analysis. We saw from the Dilly example that they talked about prospective, they talked about de-risking in the retrospective realm. And here we really wanted to use the prospective example of saying there's a need for a CIVM tool to address potential cardiotoxicity liabilities prior to first-in-human dosing. And so human-relevant data using HIPSC cardiomyocytes can provide non-clinical data to support a clinical trial, especially in combination therapies which are not regularly tested in animal models. So this actually is an unmet need because this would not be done in an animal model itself. So this may be a spot where only a CIVM can provide the usable data in a efficient fashion. Okay, so a context of use. This is our draft. So use of an HIPSC cardiomyocyte-based CIVM model is a tool for determining whether a lead investigational drug candidate, when used, A, as part of a combination with standard of care therapy, as it often happens in cardio-oncology, or B, with commonly prescribed drugs, so background therapies that are regularly used and understanding about the drug-drug interactions there. And can the use improve stratification of cardiotoxicity? And we put here a spectrum of outputs. We didn't define a single one, but you can look at cell death, toxicity, release of biomarkers, especially in cardiac troponins. You can actually look at the chronotropy as well as the cardiac conduction, which is well characterized from the iPSC data that Ksenia showed today. So that is our draft. And I'll take a moment. Does anybody have any comments or feedback or suggestions for improvement? And then I'll turn over the stage.
Any rotten tomatoes, throw them at Jordan.
I have plenty in my garden. It's starting to turn.
There's one more.
Oh, thanks. There we go. Use case. So use case A would be when the standard of care therapy has known cardiovascular risks, such in the case of anthracyclines. So think about that. It's a well-known case. So when there is known risk with the standard of care therapy, the evaluation of whether a new lead investigational drug candidate can augment that known risk. So does it increase or decrease that risk? We may not know. Usually be worried about the increased risk component to facilitate a first-in-human study. So here we're facilitating from preclinical to clinical drug development using a CIVM. Questions? Yes.
I was wondering if you also discussed potentially using this model instead of having to go back to use an animal model to show something. Could it be used instead of
So replacing.
doing additional animal studies?
Yeah.
Sometimes people, might do additional animal studies, and so this would be a choice that, instead of doing additional animal studies to demonstrate some of these endpoints, you could just do this.
Sure, sure. If we felt that the pathobiology was well validated in that system, right, and could be replicated, and you could not use an animal model to use the CIVM instead, then yes, I think that would be where you identify a signal after clinical development and use this.
Yeah,
as an
exactly.
augment.
I just
Right.
think that it's good to be able to have the replacing an animal study as a potential sort of benefit.
Yeah, we didn't think that everybody's ready to replace all animal models.
No,
for
nobody's
prospective,
replacing
yeah.
all animal, but this is an additional. This is like, if you go back and you want to do studies, and sometimes people end up conducting, I think Natalie has looked at a lot of cases, and, people have submitted up to 7, 8 extra animal studies when voluntarily without being asked. And so the question is, why would you need to do that if you can get the answer like this?
And so if we were looking at a specific conduction deficit, or we were looking at a specific cardiotoxic signal from a
Correct.
biomarker BNP, then yes, if you can recapitulate that physiology and show by de-risking, by dose modification or something in the system, it may provide additional evidence. I think that's a very wonderful point.
Okay, thank you.
Yeah, no, I think this is a great starting point with the context of use for cardio applications. And perhaps, an additional point that could be put in here is if not just a known cardiovascular risk, but if in your non-clinical studies, in your initial rat studies, you see histopathology in the heart, this may be a trigger as well.
So I'm just speaking for the group, but everyone worked really hard. We actually had a great discussion. Thank you. We came up with a lot of unmet needs in the neuro space, and then we developed a couple of them. But we developed lots of them with lack of confidence in translating animal models to humans, which goes across everything, not just neuro. But screening assays for smaller molecules crossing the blood-brain barrier. D&T is developmental neurotox on a chip. Patient stratification for neurotox potential. We talked a lot about supporting rare disease trials, but we know another group is doing rare disease, so we're just going to put our plug for all those neurology-based rare diseases. We think that there's some use for CIVM models in that space. Seizure predictability, effects on respiration, like brain-related, like CNS depression, and then disease model, blood-brain barrier. So the first one, context of use that we actually went and spent more time on developing was seizure predictability, and then assessing neuroactivity. So the context of use that we came up with was the use of brain CIVM models from humans to predict potential clinical seizure risk. And this is, something that we think would be of importance to the regulatory field because seizure risk is something that is not cannot be monitored in the clinic. So we would want to know that upfront with an IND submission. And then the second part of this was then to de-risking seizures observed in animal models to determine if there's a low level of clinical risk. So in this case, we would have models from various species. So this is the case where you see a seizure in a rat or you see a seizure in a dog study, and we want to know how clinically relevant is or is not, what is the level of clinical risk in that one. So the use case we came up with was also a species-specific seizure response is observed in a particular animal species, and then the CIVM model would be a tool for determining if there is a unique species-specific toxicity that is unlikely to appear in humans. We went for brownie points, and we did another context of use. So our second one was risk assessment of neurotoxicity caused by blood-brain barrier crossing of therapeutics. So we thought about this one. We actually have a lot of ideas, and one of them was developing CIVM models of healthy blood-brain barrier models, and then actually comparing those to models, blood-brain barrier CIVM models from the previous group mentioned special populations to identify vulnerability differences. So we're talking about blood-brain barrier models maybe from pediatrics, geriatrics, or actually disease-specific models, Alzheimer's, Parkinson's, anything, and see if there are any differences there. A context of use we came up with as use of brain CIVM models from humans for evaluating risk assessment of neurotoxicity due to unintentional crossing of the blood-brain barrier. These are your therapeutics that are not anticipated to cross the blood-brain barrier, but yet, you end up seeing some type of neurotox findings, and you want to know why. And then a use case was actually submitting an IND for use in special populations here, pediatrics or geriatrics, and the CIVM could be a tool for determining the risk of the drug crossing the blood-brain barrier in that special population, potentially causing a safety signal. Thank you.
I wanted to just ask for the blood-brain barrier thing. Do you think, for some oncology drugs, they do actually want it to cross the blood-brain barrier to treat? So could the same model then maybe be used as like a, pharmacodynamic marker for actually showing that it gets to the brain?
Absolutely.
Yeah.
You just came up with a third context of use.
Okay.
Thanks.
All right. Okay, who's here from the GI gut? The green arrow. Yeah,
Perfect. Hi, everyone. So first of all, thanks to our group. I thought we had an excellent little team, great contributions from everyone around the table, and even the people not around the table, which I was quite impressed by, that we got input from across the world. So we explored a few different unmet needs for GI tox, or GI in general. The main one we focused on was GI tox, and in particular in the space of the example of oncology, where dose-limiting tox becomes an issue and pauses and slows clinical development. with all the classical visible symptoms that I'm sure everyone in this room is aware of that people get in those trials. And therefore, we potentially need tools to support that clinical development to make those clinical trials become faster and allow us to look and determine those mechanisms of tox and potentially then explore different therapeutic regimens or administration routes for these therapeutic strategies. We also sort of delved a bit deeper into how these things could be extended further, and we discussed a lot about the incorporation of the immune system, the microbiome, and even the nervous system to kind of complement and expand these models. But for the majority of for this, we thought we'd exclude them from the unmet need. They're potentially a topic for future sessions for the complexity of the models that would need to be built. Another unmet need area that we looked at was the ADME space and the absorption of small molecule oral drugs across the GI barrier, and can we get increased confidence on the absorption of drugs before first-in-human dose studies to enable, again, them to be more efficient and go faster. And particularly, this is the case for drugs where the therapeutic index is going to be known to be relatively tight. So can we get a better prediction of what the oral bioavailability is going to be, so actually we can get, so we don't have to do so many sort of dose ascending early clinical studies, and we can get much faster to understand whether the drug is going to be efficacious and useful. So we looked and thought about quite a lot about what the performance criteria for one of these models would be to model the GI tract. There's obviously multiple areas of the GI tract that would potentially need to be modeled. And one of the key criteria that came out was the need for stability. The GI tract, I know, is rapidly turned over in a human, and so we really want to produce a model that has some ability. We would need a model that has some stability, would potentially enable it to be turned over. I think it's common to most of these contexts of use, we do need really good control compounds for any kind of qualification and validation. And then we talked through a number of key different endpoints, sort of cell viability. Some of the clinical biomarkers that are out there, and a couple were identified by the group. Particularly, I know the use of barrier integrity and mucus production, and actually potentially cilia and peristalsis would want to be looked at. And obviously, if we're going to, for the ADME space in particular, the presence of transporters and metabolic activity was key. And then the other thing that, again, I think goes across all of these CIVM tools is they do need to be compatible with the physicochemistry of the drugs that they're going to test. That's a kind of core remit. So the context of use statement we sort of honed in on after a little bit of work was that we were trying to determine and assess observed mechanistic causes of dose-limiting GI tox observed in the clinic, and efficiently then looking at comparing different dose regimes or formulation changes to allow them for efficient updates and moving forward with trial design. And this would be therefore used to determine anticipated causes, also of known drugs, so it could be used in some of the examples we've already heard about over the last couple of days with the liver. It could be used in examples where if you're going into a known class of drugs which knows to have GI tox, then you could also use the tool in that preclinical space as well. And actually just thinking about, I was sitting here listening to Jordan, and I thought the point that he made about combination therapeutics is also key. So I think is another context of use that we could add to this, is that when these, I know, particularly in the oncology space where they're using these combination therapies or there's already standards of care in these patients, I think we could add that in too. When they already know they've got GI risk, and they already know they've got GI tox, can we understand what that will look like in a co-dosed situation? Thanks.
And now the kidney group.
There you go. So thank you, everybody who was in the session. We had lots of great discussions with people coming in as well. So this is an area that where I think there's a lot of interest. And so just, based off of the context of use and what those key questions are, Some of those are really CIVM as tools for identification and mechanistic assessment. And sorry, some odd words on the slides there. But really this is really being predictive and looking and trying to identify and using those models across the various different species, and then also looking mechanistically, if you see something in the clinic, looking backwards and trying to identify what the mechanism was. So those molecules could potentially, I would say, either move forward or adjust doses. A second was CIVMs as tools to aid in selecting humans in vivo drug dosing for efficacy. And this was a lot of the discussion, I would say, that happened in our session, is really how can these be used to select those doses for patients. And then third was to enhance the support or support the concept for efficacy, and then enrollment in clinical trials of these investigational agents. And these are high overarching concepts. There. Okay, so we generally selected two, one for safety and one for efficacy to move forward. The first one was really as a tool for identification and mechanistic assessment. If we know there is a nephrotoxicity associated with the molecule, and really that was to be able to understand what that is clinically relevant exposure. So if that model can look and identify what the PK/PD is within that model, then these can potentially be used, understanding what the mechanism is within that model, to be able to help identify what doses and how to mitigate that risk for patients. So if you have the characteristics that you've identified within that model, then you'll hopefully be able to identify the patient population moving forward with, to be able to move forward with that in the clinic and get the right doses and for the patients within the clinic. We thought that was a big risk there. And the second one was really efficacy, and really looking at, for example, cystinosis, and that is a rare disease and it predominantly affects children, and it's very challenging to obtain data because you're using the data in adults to really support the proof of concept to be able to address this prospectively. And so having these tools and the added benefit of being able to have a molecule where you can evaluate this and then understand, can you deliver these compounds to, say, pediatric patients or adults, that's a big, I would say, unmet medical need to be able to do that. And having these PD, PK parameters and understanding and having these models that have these genetic diseases in them will allow us to really be able to identify what, patients and how best to be able to treat and identify, the molecules that will be able to move forward with and what doses they will be given. So really trying to utilize this to select either doses for, proof of concept for early phase 1 or even phase 3. Based off of the discussions that we've had, there's a big gap in being able to have that understanding, say in phase 1 or phase 2, and then what do we need to get into phase 3 for those patients. So I think these models, based off of our discussions, and what's currently available now, so say, what's ready now to just need a little bit more to be able to move into the clinic, it's sort of right there at the low-hanging fruit to be able to move forward and hopefully be able to help select doses for those types of patients.
All right, Jay.
All right, so again, thanks everybody for a great discussion, very efficient discussion. Appreciate it. So we were given a very broad topic. So the first thing we decided to do is figure out what is the premise of what we were going to talk about. Generally, we also divided it into two classes, rare diseases and larger population disease models, okay? And the unmet need was, for rare diseases, generally no animal model or limited clinical data. Large scale diseases, confounding or limited animal data, or questions arising from clinical observations. So those were our unmet needs for these two different classes. And again, we wanted to also not limit it to mechanistic, target-based, but also use phenotypic models. We also, efficacy models did not have to go through ISTAND. There are many other avenues through FDA, so we wanted to make sure that was understood. And again, we believe that rare disease COUs can be made distinct from large population efficacy model COUs. And then rare disease, and then basically use the parameters from the rare diseases to then expand upon them to the large populations. So we also figured that the best thing to do for the community with this is just identify what the different parameters that would be needed, okay, to generate COUs for rare diseases. Genetic basis or serum application, stages of disease, early, middle, or late, treatment of symptoms versus disease reversal, what endpoints are being utilized, either could be your functional and or biomarkers, and then how to quantify these biomarkers, disease etiology, clinical manifestation, sex, cost, and timelines. We thought that basically somebody could take one variable or a combination of these variables and then build a COU around it, okay, for a rare disease, okay? And for large populations, we basically, the premises usually have more information from either animal models or clinical etiology than for rare diseases. Context of use variables, first we would take all the things from the rare diseases and include those, and additional factors for large populations, comorbidities, lifestyle environment, heterogeneity, whether you wanted to focus on a small population, say for diabetes, or try to look at something that was overarching over all patients for something like diabetes. Okay, and then also patient stratification. Okay, so you could basically choose whether you're doing type 1 diabetes, type 2 diabetes with aged or younger, in terms of stratification. So again, somebody could just sort of take this list and then say, okay, what am I doing, and pick out those portions, and then develop the context of use from that, those variables that they picked. And last, for our specific COU is for ALS. Regulatory elements characterize the phenotype or genotype relationship, preclinical, and variability of drug response, preclinical phase 1, phase 2, to support patient selection in clinical trials. So the actual COU we would submit would be functional model used to characterize phenotype and genotype relationships in order to select appropriate patient populations for the study, would be the COU we would propose. Any questions? All right, thanks.
All right, well, thank you everyone for, I think, an excellent exercise in going through and looking at questions of interest in different organ areas, as well as developing an initial COU and going through that practice of something that's regulatory facing applications with complex in vitro models. So thank you very much for all the moderators and the participants in these sessions today. And I believe we have one last piece for today's meeting, workshop to close out the day. And if Nicole would come up for a moment, she's got a few comments.
Thanks, surprise. Thank you for letting me just slide in there real fast. For those of you who haven't met, I'm Nicole Kleinstroyer. I'm the director of NICEDM and the executive director of CIVM. You don't need to remember what those stand for, but basically we're the U.S. federal resource for alternatives to animal models. And we support basically every federal agency, particularly our regulatory partners like FDA, in the space of NAMs. And at the beginning of this year, in March, we, the CIVM Validation Work Group, published this guidance document on validation, qualification, and regulatory acceptance of NAMs. FDA co-chaired the VWG, and we had representatives from, I think, 5 different FDA centers as part of the work group that drafted this document. I think it's really, really useful. I encourage you to read it. In an effort to operationalize the concepts in the validation guidance document, we've launched an event called the CIVM Method Developers Forum. And so this is to try and create the opportunity for method developers to connect with end users, predominantly our regulatory decision makers, but also folks in regulated industry who are interested in using some of these complex in vitro models and other types of NAMs in their development pipelines, and to try and encourage communication between method developers and end users early and often in the method development process. And we are focusing them around different endpoints. So the first one we actually just held last month in August. It was focused around carcinogenicity. The next one, which will be early next year, is going to be focused on cardiovascular toxicity. We will also then be holding MDFs for neurotoxicity, for developmental and reproductive toxicity. The list is basically endless. And then once we finally get to the end of that endless list, we'll just start over and go back to the beginning. And so the way that we're approaching this is we're actually asking our agency partners to pre-record presentations that summarize their information needs and their decision frameworks and how they assess particular endpoints or toxicities of interest. So what is their current approach to cardiovascular risk assessment, and what types of data are they currently using for that? Where are there opportunities for improvement, gaps that could be filled by more human relevant NAMs, et cetera? And then those pre-recorded presentations from our different agency partners are put up on the ICVM website. So if you want, you can go to the website right now and you can see 8 different presentations from FDA CDER, CIVM, EPA's various program offices, CPSC, NIOSH, OSHA, et cetera, specific to carcinogenicity, which was the first topic. Right now, we're asking people to volunteer to pre-record presentations around their cardiovascular risk assessment needs. And then we ask the method developers to view those presentations and to think about how their methods might adhere to or might fit into decision frameworks or information needs or particular contexts of use that have been outlined by our federal partners. And then we ask them to structure their presentations based on the key concepts that are in this validation workgroup guidance document. So things like, how has the method been technically characterized? What is the intended context of use? How have you demonstrated biological relevance, et cetera? So I just wanted to make you all aware of this event. The website is right there, and of the guidance document. So hopefully this, I think it dovetails really, really nicely with some of the conversations that we've been having the last 2 days. So thank you for giving me the opportunity to jump in.
Thanks, Nicole. I think that is going to be a nice resource and discussion point moving forward in the field.